5. Just wanted to, as you saw in the chat, if you've got it open, I wanted to point out that I was realizing, and this is not unusual at all for me, that we are going a little slower through the innate and immune system and the adaptive immune system than the syllabus has us going, and I don't want to fall too far behind. So what I did was this morning I edited the groups for the lecture questions, and what I decided was, since I provided that table all filled out for you, that the people that were working last time on lecture questions 14 and 15 that just ask you to name members of the table that are made by the liver and what they do, and then ones that are released by white blood cells and what they do. Well, that's all covered for you in the table. I think you have that fairly well established. So what I'd like to start with is the finishing up the complement actions, which we didn't cover. I think Noah really nicely summarized the 3 ways you activate complement. So I think we finished that first part of that group's question, but the second part has to do with once you've activated it and you make C3, what happens next? So that part and the alpha and beta interferons, I think, were in the same group. So alpha, beta, and gamma interferons. So those 2 lecture questions, if you were in that group, and I think Noah was the spokesperson, but if there was another spokesperson for the other questions, then please let me know, and I will put you back into that group. And then the last question under the innate lecture questions has to do with natural killer cells. And this is actually the beginning of, in the beginning of the next lecture question, the second lecture question. So I put that one as its own, if I remember rightly, I put that as its own group last time. And we'll just leave, if you were in that group again, let me know if I actually assigned that group. I can't remember. Otherwise, I'm going to join that question to questions 3 and 4 of the adaptive. And that's because of the discussion of MHC1 and MHC2 that we began last time. So that means I'm skipping lecture questions 1 and 2 of the adaptive. And the reason I'm doing that is that we've already covered that material. It's kind of a review of a comparison of the innate and the adaptive, and then a review of B cells versus T cells. And I think we've covered that pretty well. So if you have questions on those ones we're not covering, just of course, bring that up. So otherwise we're going, other than those changes, we're going to go forward. I'm going to try to get through the second essay question, so you have something to work on, and then we still have quite a few questions ahead of us. There are a lot of lecture questions in the in the adaptive. So on Thursday we'll still be doing the adaptive, so we will still be behind. So please put in chat if you were in the group on complement, which was from last Thursday. Otherwise, I will probably see if Noah wants to finish up at least the C3, C4. And if I did assign someone to group G, then let me know that you're in that group. And we're going to reconstitute that, but with the new lecture questions, which I'm calling group B, on the adaptive. Okay, so I also want to apologize. I don't know what happened. You realize there has to be like 3 steps. You probably don't know that. For me to make this recording, transfer it, upload it into the program that Canvas has for uploading these recordings, and then from there upload it into the discussion. So somehow that lecture got lost, but hopefully you had a chance to listen to it since I put it up on Sunday on the material that we're going to be covering today. So we still are a bit short on folks arriving. I only have 24 people here, but I'm going to go ahead and assign you to your groups. So let me get into the breakout rooms, and it looks like I think I have 6 of them. Create, and since whoops, why are these already here? I don't know why that's not started. Okay. How do I get rid of this? Recreate, maybe that's it. Oh, well, maybe since they're already here, I'll just rename them. I don't want to delete all of them and send you into this rooms. So I'm going to call the first group, which is going to be Jasmine, Jeanette, and Jessica, and Sarah. I'm going to give you guys the group G and B questions. That means you're doing the part on the natural killer cells and those first 2 questions. Well, questions 3 and 4 that are in the adaptive. So group G and B. Again, double check your announcements if you're unsure. And then the next group has Augustine, Lewis, Sana, and Sophia, and we're going to call you group H. And then the next group, which is Danielle, Dina, Pradeep, Stephanie. I hope these people are here now that I'm thinking about it, because I think this is something remnant left over from last time we had a lecture. We're calling group I. Do I even have that group? Yeah, let's call that group I. Put that on there or not. So you're doing lecture questions 6 and 7 from the adaptive ones. And now then, so we're going to have Alexis, Eric, Kaylin, and Selena work on lecture question 8. I hope I'm matching my posted groups, IJ. So I'll just put lecture question 8 on this in case. In fact, the last one, since I'm confused now, we'll put lecture question 7, I believe it is. 6 and 7. 6 and 7. Yes. And J. So now we'll go back to, let's see, we have Angela, Brian, Reed, and Noah. Since Noah was in that group, we're going to go back to the complement ones. And that name was group E. Group E, and I'll say innate complement. Okay. And that still leaves one more group. So let's give you, yeah, so this is, I forget the letters I had on there, but group JK, I guess. And you're going to do lecture questions in the adaptive 9 and 10. 9 and 10. Okay, so that's Alex, Jennifer, June, and Stacy. So let me see if I have anyone unassigned, and I'm going to open those rooms. So going back to complement, are you going to be the spokesperson again, Noah, for finishing this up, or is someone else in that group?
Manreet will handle it, and I'll handle the next question after this.
Okay, all right, Manreet. So I put down on the screen here where we had left it off. I don't know if you want to recap it, or I can, whatever you desire.
I can maybe go over it, yeah.
Yeah, just briefly, since it's something we've covered. Okay.
Okay, so we have the classical pathway, which is activated by antibodies coding target cells, and it is specific and is the best in secondary infections or late primary infections, as the antibodies need time to be made. This pathway is both cellular and acellular. Then we have the lectin pathway, which requires certain sugars on the pathogens and is activated by lectins, which is released from the liver, which needs to be activated by macrophages to release it. It is activated when lectins bind to specific sugars on microorganism surfaces. This pathway is usually cellular. And then the last one, the alternative pathway, is activated spontaneously, such as when there is a lack of inhibitors on microorganism surfaces, which allows the process to proceed, meaning that the complement is being activated.
Brilliant. That's a nice summary. So you can see, since there are so many different ways to do this, it must be important. Usually when there's a lot of redundancy, it's both because it's important, but also because microorganisms come in a wide variety. So you have various methods to cause activation of complement. So once you get complemented activated, what happens next?
Okay, so you would create C3A, and that enhances inflammation via binding to mast and basophils. And hold on, let me just get to my notes as well. Okay, so that will stimulate an increase in histamine levels, while B3B will increase phagocytosis, and by doing opsonization, which binds pathogen surfaces, which enhances phagocytes even if there is a capsule covering the pathogen's PAMPs. While another mechanism that C3B also does is it joins with MACs to make a MAC attack, which is when a complex is placed into the membrane and creates holes, which is lysis, in the membrane, causing interstitial fluid to leak out and thus kill the pathogen. So a way to memorize this is the acronym OIL. O stands for, I believe, opsonization, and then I stands for inflammation, and L stands for lysis, which is the holes in the membrane with the MAC attack.
Great. So the one thing I will ask, if this is a short answer on your, and I'm asking you to explain complement's effects, then don't just write OIL. At least explain what OIL means, and make sure you know that correlate C3A with its outcomes and C3B with its outcomes. So this is going to be an enhancer of your innate response, and in that way indirectly kill pathogens, so by inflammation and opsonization. So the only direct antimicrobial is really this MAC attack. So this would be pointed at a cellular type of pathogens, but inflammation could increase phagocytosis of acellular type of pathogens. So it's a broad spectrum, meaning it goes after lots of different pathogens. Okay, so complement, important stuff. Don't worry about, like I put and others here, knowing all the lettered complement subunits that you need to make a MAC attack. So I'm not worried about that. And remember that up here, you're combining these things with complement subunits. So I didn't write that here, but each one of these requires, again, don't worry about any numbers. different complement subunits for them to come together to make C3. Okay, so group E, I keep changing this poor group. I think I also gave you the alpha and beta interferon question, right?
Yeah, you did.
Okay.
So how do I go about this? Alpha and beta interferons are basically produced by an infected cell, and they release interferons and send it to their neighboring cells, which allow them to produce like an antiviral protein to block the virus. And it also signals the activity of natural killer cells to kill the infected cells.
Great. All right.
And then the gammas will increase activity of macrophages and aid in T cell differentiation. In your notes on lecture, it said that alpha and beta increased production of MHC1, but on the table it says that gamma actually increases production.
I think the way I wrote that slide was a little bit confusing. So this increases MHC1 and MHC2 expression. So in a way, as we'll see, this occurs during the adaptive, although it can help the innate. And we'll see more how it helps the innate when the next group talks about these natural killer cells. Because these particular things are released, this one almost more like a cytokine, and this one like a cytokine too, for that matter. These aren't directly killing anything, but in general, you could broadly say that interferons in general are particularly good against intracellular pathogens. But there is a broad spectrum of these as well. And the book only really focuses on this effect much in that it makes a picture of that effect. But some of these change inflammation. They have other effects that the book doesn't really go into. And I think it's whatever's in the table is what I would want you to know. So alpha and beta are really solely within the innate because they're being released by white blood cells of the innate response, and they're acting on either your body cells or released by your body cells, or they're acting on innate members like the natural killer lymphocyte. The gamma, as you'll see, are released mostly during the adaptive immune response.
I don't know.
Somebody has their microphone on. If you could turn it off. Let me see, I might have to. Well, let's go forward. Hopefully it won't unmute because we have to get the next person speaking. Okay, so that's a good summary. So let's talk about these natural killer cells and who is going to cover that. It's in the group G and B people.
I will cover number 16.
Okay.
So describe how a natural killer cell recognizes an abnormal body cell and what it does in response to finding an abnormal cell. So natural killer cells detect non-self cells by recognizing a lack of MHC1 or the presence of antibodies coding the pathogen. And they then kill the pathogen by releasing perforin and granzymes that induce apoptosis.
Great.
And then they also release interleukins that call in phagocytes and increase inflammation.
Okay, that's nice. Thank you very much. So we need to talk a bit more about this MHC1, which I think the next speaker will, or maybe the same speaker will address. What is MHC1? But this is a common way that natural killer cells know when you have an abnormal body cell. So as I mentioned in the lecture, the book really focuses on abnormal cells that are virus infected, but cancer cells also, and even cells that are no longer metabolically active for whatever reason, dying cells could be eliminated by natural killer cells. So what it means is that if a cell is not generating this MHC1 at sufficient amounts, then you're going to see the natural killer cell become activated. So they do give another way that activates natural killer cells. That would require that a body cell, because this is again on the body cell surface, would have to have an antigen on that surface. So an abnormal body cell that's viral infected, sometimes what the virus does is it inserts viral proteins into the membrane of the body cell that it's infected. And then when it goes to exit the body cell, it actually takes some of that cell membrane and coats the virus with the cell membrane. And in fact, many mammalian viruses, including the ones that infect us, have this little piece of stolen membrane on the outside, and the receptor to infect the next cell is embedded in that piece of membrane. So if you've mounted an adaptive immune response and you are making antibodies against the antiviral proteins that were inserted into that body cell's membrane, then that would be another way. You could have a natural killer cell recognize that this is an abnormal cell because it has antibodies on it. Once the natural killer cell realizes that, it releases granzymes, and these are the things that cause the apoptosis. Perforin allows the granzymes to enter into the cell by making pores. So this yields, yields pores. And the interleukins are going to increase other members of the innate, like macrophage activity. So once you kill the cell, remember these are one of your body cells, you want to eliminate whatever viruses may be inside that body cell, or if it's a cancer cell, you want to eliminate the cell altogether and munch it with phagocytosis. Okay, so these natural killer cells are our first introduction to MHC1. So the next person in this group is going to describe the functions of MHC1 and the locations of MHC1 versus MHC2. And this is a big topic. It's the very beginning of the second essay. So who is going to be the spokesperson? Maybe say your name too, because sometimes I don't recognize voices.
Professor Gyalsung, I'm taking this question.
Great.
So, the location for MHC1 is in all nucleated cells.
Okay.
MHC2 is APCs, which are dendritic, macrophage, and B cells.
Great.
And moving on to functions, MHC1 is MHC1 present endogenous pathogen that originate from the cytoplasm, meaning inside the cell, and it is an intracellular pathogen, which are all foreign intracellular antigens or can be altered self-antigens or target cells, and these are for the destruction. And MHC1 present antigen to cytotoxic T cell CD4.
Okay, and I put a 1 there for a minute, just display exogenous antigen.
Pathogen, and that originally extracellular, and it's for CD8.
Okay, great. And then the one I left out here is NK cells, right?
Yes.
Yeah, we just mentioned that they do it. And then who looks at it, who sees it, who sees, binds it.
For
And
the
you said
CD4.
CD4 is this guy, right? And this
CD4 for MHC, or yeah, and then CD8 for MHC1.
And the natural killer cells.
Yes.
Okay, great. All right. So that's what this asks about, the locations and the functions. Now, when I'm saying binds it here, where did I put that? Binds it. What I mean here is we're going to be talking shortly about double recognition. So there was that table that I kind of fumbled when we went through the lecture, since I kept getting somebody calling me desperately over and over again. Anyway, double recognition means that the MHC2, which has the self combination, which we'll probably talk about a little bit more. So MHC1 and MHC2 are unique to each individual person. And it's a combination of different subunits that are put together into this big complex. So unless you have an identical twin that's genetically identical to you, there isn't anyone else in the world with your particular combination of MHC subunits. Once these are put together, that labels all your cells as you. So this double recognition involves a protein on the outside. I think it's a protein or a glycoprotein on the outside of these lymphocytes. So if it's a natural killer cell, it's interested in the MHC1. I don't know that it does bind to the MHC1, however. But if it's a CD8 cell, it is going to bind. Its CD8 is going to literally bind to the MHC1, and it will only bind MHC1, not MHC2. So this guy binds only MHC1. And this guy only binds, this guy over here, only binds to MHC2. So we're going to pretty quickly go into the reaction of causing this binding, this double recognition to happen, is to try and trigger these immature T cells that have already become immunocompetent, meaning they've gone through the thymus, to eventually become the active types of T cells after undergoing extensive mitosis. But they can't do that without the next step. You're going to have another step involved called co-stimulation. So in a way, the double recognition is a slightly different in what they recognize, but they're fairly similar in the idea that for the CD8, you need an MHC1, and for a CD4, you need an MHC2, but otherwise they're using a membrane protein or glycoprotein on the outside of the lymphocyte, and they're actually binding it to the MHC on the antigen presenting cell. Co-stimulation is a little more variable for these different types of cells. So we are going to go into that shortly. So is there anything else you wanted to say, Kelsang, or are you?
I am okay with this question, and the other question will be answered by Jasmine.
Okay. All right. So the other question is kind of a far-ranging question. So Jasmine, it says, describe what an antigen-presenting cell does, and then name 4 types. And you'll see in my post-it, I said, well, it's sort of 3 types in the book, but we are going to talk about a potential 4th type. So if you've only come up with 3, that's okay, because I'll talk about the the fourth type. So what does an antigen presenting cell do, and who are they?
I think that's group C. So our question is, yeah,
You're right. I'm sorry. That's the next person. Okay.
is describe the mechanism by which antigens are bound to MHC1 versus MHC2 and what cells display both. Is that correct? Is that what you have?
Yes, I'm sorry. I skipped that one.
So,
So MHC1, how do you get that?
so for MHC1, we have, so say if you were a cell making, for example, keratin, the endogenous antigen would be broken down and taken out of the cytoplasm to be put into the smooth endoplasmic reticulum. From there, you'll find in the membrane of the smooth ER that you'll have an MHC1 where the broken down antigen peptide would be placed. A part of the membrane would be pulled off, plugged into the plasma membrane to be presented on the outside of the cell. So if this were a viral protein, then your CD8 would be interested in the antigen peptide, but it would have to know itself. So it would have the MHC1 to recognize that. It does this by the CD8 binding to MHC1, and the receptor would bind to the antigenic peptide. If this were a natural killer cell, the issue would be that you would see a decrease in MHC1 because you're generating those viral products. So whatever MHC1 there are would get loaded on, but due to the lack of MHC1, the natural killer cell would kill the cell. The CD8 cell would bind to the MHC1 molecule, and it would have the targeted antigen, and it would kill it. That's what we have for him.
Okay, very nice. All right. How about the MHC2?
So for phagocytosis, I'm assuming it would engulf the bacteria. It would take it apart from its phagosome, join into a lysosome, and break it up, which would lead to some of the antigens to be placed in a vesicle, which then fuses with the vesicle with MHC2, and the antigenic peptide would be loaded on. The loaded MHC2 molecule would then be placed onto the membrane and would look for a CD4 cell. The CD4 binds to the MHC2, and the T cell receptor finds that particular antigen. If the CD4 cell targets both bound as well as the correct interleukins, it undergoes mitosis at a high rate, which allows it to make multiple copies of itself, and it becomes T helper cells and T regulatory cells.
Okay, again, very nice. Now, one exception to this rule that we made a wall right here, and that's dendritic cells. And this is something that the book is quite confusing about. I think it's poorly dealt with in the book. It shows a picture, but doesn't explain how it got there. So in the case of dendritic cells, if this were a pure macrophage, not a dendritic cell, and it's going to be an antigen presenting cell, then it would do this process. And all you would get was an antigen presenting cell with the MHC2 antigen on the outside. If this was a dendritic cell, what it can do is take this phagosome and make it into a lysosome, fuse it with a lysosome, make it into a vesicle that contains antigens. And by means that I have no idea, perhaps it enters the smooth ER, perhaps it just it makes vesicles with both MHC1 and MHC2. The dendritic cell can take this and put antigens that were obtained from this, well, maybe I should put it from over here, from this phagosome and put it on both MHC. So this is for a dendritic cell. So it can have an exogenous antigen on MHC1. It can have an exogenous antigen on MHC2. And this is one of the things that makes these unique from the macrophages. And even though the macrophage can be an antigen presenting cell, it's going to have a hard time activating the CD8 T cells. So when we get to that part, which is a little ways off, you're going to see that, again, the book doesn't deal with it very clearly. It kind of skips over it in the text very briefly and then shows a fairly confusing picture. So I do want to spend some time when we get to that part. All right, so so good job, you guys. Let's go on to H, where I started to introduce who are antigen-presenting cells and 3 types of them, and then we'll talk about a potential 4th type. So who is the spokesperson for Group H? You only have the one question.
So antigen presenting cells, the engulf antigens.
Is there a spokesperson for Group H?
Can you hear me now?
I'm sorry, who is this?
Hello?
I can hear you better now, Elena.
Oh, now I can't hear anything.
Got their microphone on, I'm not sure.
There. Is it working now?
Yes.
Okay, perfect. Sorry about that. So antigen presenting cells engulf antigens and present fragments of the antigen on their surface so they can be recognized by T cells, which will then bind them to their CD4 receptor. And 3 types of antigen presenting cells are dendritic cells, macrophages, and B lymphocytes.
Okay. Okay, so engulf is true for these 2, right? But for the B lymphocyte, they're going to do endocytosis. Because what's going to happen, and again, once again, the book was a little bit, I think it was in the words, but there were no pictures for this. The B lymphocyte is going to have a receptor for the antigen on the outside, which will trigger endocytosis. And that receptor is going to be highly specific. It's going to be an antibody. Versus these 2 guys who use their PRRs to know who to engulf, and they do phagocytosis. Maybe I should have put that over here, phagocytosis. And they use your PRRs binding to PAMPs. This guy is going to use an antibody to know who to bind to and then do endocytosis. And once they do, all of these guys are going to be putting on MHC2. But what did I just say about these guys? They can also load it onto MHC1, right? And MHC2. Now, why do we need it on MHC1 is it will be a way to activate the CD8 cells. So when you go back to looking at the MHC1 versus MHC2, remember that, oops, maybe I did it up here. Yeah, who sees? This CD8 cell can only see an antigen if it's on MHC1. This one needs MHC1. This guy needs MHC2 as an antigen presenting cell. So this one needs antigen on the MHC1. As an antigen presenting cell, and this guy needs antigen on MHC2. So if we want to get to become an antigen presenting cell, if we want to get the CD8 T cell activated, then we've got to present the antigen on MHC1. Is that clear? Any questions about that? Okay, Ileana. So once you've got these guys being presented, that's where the double recognition is going to undergo the beginning of trying to get your activation of your T cells or your B cells. Later, we'll talk about those. So number 4 in here, and I would agree with most books, they don't generally call these antigen presenting cells, But if you are a body cell that's infected with a virus and you put a viral protein, or if you have cancer, a cancer protein on the outside, an abnormal protein on the outside of your cell, you're going to present that on an MHC1. So even though these are not really considered antigen presenting cells, this is another way that the CD8 cell may encounter its antigen. So I'm going to put it as number 4, a body cell with antigen, and this is not a self antigen, but a non-self antigen, an abnormal antigen on MHC1. So one of the reasons you have many more CD8 T cells than you have CD4 T cells wandering around in your blood, it's usually about a 2-to-1 ratio, is some of these CD8 T cells are looking for areas of inflammation where they will leave. And if that is inflammation due to, say, a virus infection or a cancer, then those cells will potentially encounter their MHC1 with their antigen on a body cell. They have then had the first step. This would lead to double recognition happening. Now, remember from what I said earlier, in order to fully activate these cells, they have to have not just double recognition, they have to have co-stimulation. So we will come back to that in a little bit, talking about how these CD8 T cells become co-stimulated. So right away, you're seeing there's 2 ways that the CD8 can get double recognition. One way, and I'm making it in purple because we're following the MHC, one way they can have double recognition is some dendritic T cell can show them their antigen on an MHC1. Another way the CD8 can have, that should be CD8, double recognition, is this down here where the body cell shows it. So I'll put of CD8 T cell. Is that clear? I mean, this kind of makes sense, right? You want to patrol your body for places where infection or cancers may be occurring. CD8 T cells are going to become the T cells that can kill body cells that are abnormal. And they're only going to kill abnormal body cells that present the antigen on the outside of them. So they're going to be very precise, unlike less like the natural killer, which are not all as precise. They're just looking for whether or not there's an MHC in enough density. These guys need the MHC plus the antigen in order to target them. So they're real. So you can think of natural killers as like a heat-seeking missile that's anything hot is going to kill. They're going to kill. Whereas the CD8 T cell, when it becomes a cytotoxic T cell, has one specific target in mind, the antigen on the MHC1 being presented. It's much more honing in on a particular specific target than anything that's hot. I'm not enough of a military person to know what kind of missiles would be that precise. Are we good on that? All right, so the one I have in purple here is not really discussed in the book, but I thought because it does make sense to think about how you get rid of viruses in your body, it's probably worth knowing something about that. But in general, they don't classify these as an antigen presenting cell, even though in a way double recognition is going to have to happen. Okay, so I believe H, I just gave you guys that one question, right? Because the next one is kind of a topic shift, and it goes back to talking about immature T cells as they leave the bone marrow. I do think we covered some of this, but let's let's see who is in group I that is going to be our spokesperson for this.
That's me, Dr. Kaufman, Dina.
Okay, Dina. Okay.
So question 6 is, what 2 conditions must an immature T cell meet to survive selection in the thymus? The first is positive selection. Only T cells that are able to recognize the self-MHC proteins will survive to move forward to negative selection. And then negative selection is the second condition, and this ensures that the surviving T cells do not recognize self-antigens displayed on self-MHC. If they do, they are eliminated by apoptosis. Negative selection is the basis for immunological self-tolerance, ensuring T cells do not target and attack healthy body cells, which would, if they did, that would lead to autoimmune diseases.
Great. Okay. All right. So you want to get, when we talk about cytotoxic T cells killing your own body cells, then you want to make sure that you're not sending these killers out and they're doing damage to your own body. Like Dina mentioned, if they are, and in some cases, that's what happens in autoimmune diseases, where your cytotoxic T cells are going out and damaging your own body cells because they're mistaken in recognizing an antigen that is not an antigen itself. Not a antigen, really, it's a self-antigen. So it's nothing foreign, it's part of you. So if you can't pass either of these, then these guys die. So once you go once they migrate into the thymus, there's hormones involved here. And I don't know the particulars, whether they need a hormone to even start these selection processes or the hormones are used during the selection process. But there are 2 hormones involved. Don't worry about their names. I think they're actually thymopoietin is one of them, which kind of makes sense. But anyway, you want to hone it down to just the T cells. Now, I focused on the CD8 T cells because that's more obvious why that would, if you had an inappropriate response of those, that would be bad because it would be killing your own body cells. But if we have an inappropriate CD4 T cell becoming helper T cells, then you may end up getting activation of your innate or exaggeration of innate responses that can also do self-damage. So we will shortly get to the helper T cell. That's where we're going briefly next, and talk about how those helper T cells act. Okay, so once they leave, they are now immunocompetent. And let's see if the next question is to compare and contrast the CD4 and CD8 T cells that are leaving. And I name them CD4 T cells and CD8 to try and indicate that these guys have not seen an antigen presenting cell. The book tends to just call them all, T helper, T, but they don't become a T helper or a cytotoxic T until they've undergone clonal selection. I think it's clear about that, but they just call them a T cell. I just thought it'd be clearer for you guys if they're just totally immature, they're not going to do their actions, then we'll call them a CD4 T cell versus a CD8. And that's pretty common in the literature, how they're referred to. So if we compare and contrast them, besides them being similar that they're T cells clearly and had to do negative and positive selection, is there other things? Is that you still, Dina?
Yeah, that's still me.
Okay.
So I've got that the activation of CD4 T cells results in the clonal selection, primarily of helper T cells, but also of memory cells and a variety of T cells, including regulatory T cells. CD4 T cells require MHC2 and exogenous antigens on the dendritic cells to differentiate. The activation sequence begins with double recognition and subsequent binding to the dendritic cell's MHC2 antigen complex. The release of interleukins between the dendritic cell and the CD4 cell results in co-stimulation. Additional interleukin secretions results in B7 membrane protein secretion in the B7 membrane protein secretion needed in CD8 activation. Clonal selection occurs once double recognition and co-stimulation trigger the cells to proliferate. The primary product of the cloning of CD4, I already said that, never mind. The helper T cells are required for the activation of both CD8 and B cells. Activation of the CD8 T cells results in the production of memory cells and cytotoxic T cells. When the CD8 meets the MHC1 and self-antigen presented by a dendritic cell, double recognition occurs. Both begin secreting interleukins as seen with CD4 cells. However, co-stimulation requires the additional interleukin secretion by the B7 membrane protein of the helper T cell. Co-stimulation, as seen in CD4, triggers clonal selection and differentiation to their end products of memory cells and cytotoxic T cells.
Okay, so on my graph here, this one forms T cytotoxic and CD8, right? Memory. And then let me catch up with you. Fell behind. This is the MHC1 antigen, and that's for double recognition. Okay. And this one also, and this is where it's going to get a little muddy, but it can use interleukins, like she said, from the T helper. And that will lead to, so double recognition here. We have co-stimulation. This is for co-stimulation. And that leads to clonal selection. Okay. Am I getting everything, Dina?
Yeah, I think so.
And that leads to T cytotoxic plus the T memory, which releases perforins and granzymes to kill infected body cells. Now, in order to kill those infected body cells, let's put this down. When body cell displays MHC1 with the antigen. So that's where we're talking about it being very, very narrow in its targeting. So that's almost another double recognition, although they don't call it. You're going to have the CD8 bind to the MHC1, and you're going to have the antigen receptor bind to the antigen. When that cytotoxic T cell reaches the body cell, you're going to release perforins and granzymes, just like the natural killer cell did, and that will end up killing that body cell. Okay, great. So I think we jumped into some of the material in the next group's question, which talks about CD4 undergoing this double recognition. I believe that group is, I forgot the letter. Who's doing a lecture question? I think it's IJ. Who's in group J doing lecture question 8 for the steps for an immature CD4 cell?
I'll be doing lecture question 8.
Okay.
So the question was outline steps for an immature CD4 T cell to become activated, co-stimulated, and undergo clonal selection. So the bacteria is phagocytized now and is inside the phagosome and fuses with a lysosome, which creates residual bodies and are bound to MHC2. Eventually, you load many of these antigens because it is a bacteria onto the dendritic cells, which is an antigen-presenting cell. The dendritic cell will run into lymphatic tissue and look for CD4 cells because you do not have many T cells that are specific to that antigen. The T cell is just waiting around for the antigen to bind, and when it does, double recognition occurs. First, double recognition occurs because one CD4 protein binds to MHC2, and the second one is T cell receptors binds to its specific antigen. Like fitting a key into a car, now you need gas in order to start the car. So you need co-stimulatory molecules, and there is exchange of interleukins. When binding occurs, the dendritic cell will release interleukin 1, and the other cell will release interleukin 2, which causes both cells to be changed. They're tracking the change of CD4 cell and undergo clonal selection and make a population of many T helper cells, which are interleukin secretors, and a few memory CD4 cell, which can work more rapidly and help with secondary response in case you run into the same antigen. Also, T regulatory cells are produced from the CD4 cell.
Great. Perfect. Okay. So then we get T helper, T reg, and CD4 memory. Now remember, when you do clonal selection, you're not making one cell, you're doing a massive amount of mitosis. So you're getting lots and lots of T helper cells. So I'm going to say the most here, most cells. And there are some T regulatory cells. The book doesn't talk much about T regulatory cells, but stay tuned to these guys because they're already being used therapeutically. Because they end the immune response, they are used therapeutically to suppress autoimmune conditions. So remember, autoimmune conditions would be when you mount an immune attack on your own body tissues. So what they do is they actually extract these lymphocytes from the bloodstream of the individual, or of an individual, probably an animal, I guess, but no, I don't know where they get them, sorry. But at any rate, they get these from that individual, they do. They get some and they trigger them to undergo mitosis, so they build up a lot of T regulatory cells, and that T regulatory cells against that particular self-antigen that's being attacked, and they've actually given them back to the person, the same person they got them out of, and suppressed an autoimmune symptoms from this. It doesn't cure the person, it's just suppressing their symptoms at that point. So they don't say much about these guys, but both of these, as Selena said, their job are to release many different interleukins and other cytokines, which we won't name or worry about. So you just need to know that interleukins are released by these guys. That's all they do. They are sitting in the lymphatic tissues, secreting these interleukins. They don't travel out and go find the pathogen or anything. They're releasing signaling molecules to enhance other aspects of the immune system. Now, one of the reasons they are staying in the lymphatic tissues is because they're going to be important for activating B cells and CD8 T cells as well. Remember, there's fewer of these T reg, but you still make more than one. So it may depend on the particular antigen, but maybe, I won't put many, but greater than one CD4 memory cell. So if you thought about this being a library, it's like your T cell library has one copy of every book when you're born or shortly after birth. And that's what's being generated by your bone marrow before you've seen any antigens. Once you see an antigen and they undergo clonal selection, T cells or B cells generate memory cells in greater numbers. So you don't have one copy of that book anymore in your body. You have multiple copies of these books. And these memory cells can be long-lived. They don't necessarily live, all of them, for your whole lifetime. And they then what that means is that there are more cells that recognize that particular antigen than you had if you never saw that antigen. So when we talk about the secondary response, it's going to be faster, probably in part because you have more memory cells in your library than the original, than before you saw that antigen with the primary response. Okay, any questions on that? Let me see how much time we got left. We've got some time left. So, so I believe it was briefly talked about, but is somebody doing 9 where we go into more detail about what these T helper cells do?
Yeah, I'll be explaining that. It's Stacy.
Okay, Stacy, great.
So T helper cells help amplify the responses of the innate immune system, and they do this through such as activating macrophages that are more potent killers. And when more and more cells are released, it generates a momentum in the immune system, in the immune response. So in the humoral immunity, the helper T cells bind with the non-self complexes of B cells that have already encountered its antigen and is already displaying it on the class MHC2 on the surface. The helper T cell then releases interleukins as co-stimulatory signals to help complete B cell activation. In the cellular immunity, the helper T cell binds to a dendritic cell, which then helps the T cell stimulate the dendritic cell to express co-stimulatory molecules. The dendritic cell can then activate CD8 cells with the help of interleukin 2, which is secreted by the helper T cell. And it's required for clonal selection, and helper T cells also stimulate, I believe it's T cytokine cells, and it also activates natural killer cells.
You mean cytotoxic T cell? Is that what?
Yeah, sorry.
Yeah, increased natural killer cells activity. Okay, there's one more thing I want to add to that, and that's some of these interleukins are colony stimulating factors. So they're going to act on the bone marrow, so that you can increase the rate of leukopoiesis, but making more white blood cells. So that's great. They do increase macrophages. Some of them are pro-inflammatory. Sometimes they're just as Stacy brought up, that by amplifying how these guys act, you end up seeing more inflammation because they release more leukotrienes, et cetera. So it's an amplifier of your innate response. So I wanted to just, as a side, mention that, of course, you know about HIV/AIDS. What you might not know is HIV/AIDS targets these helper T cells and kills them. So normally you have and I said that wrong earlier, I think. Normally you have about twice as many helper T cells in your blood as the cytotoxic. I think I said it the other way around, so please forget what I said earlier about that. So normally you have a CD4 ratio in a normal person to a CD8 would be about 2 to 1. And as the person progresses into AIDS, having symptoms, what happens is the HIV, which infects the helper T cell, begins to eliminate these helper T cells. And you see they monitor the progression of AIDS in an individual often by looking at this ratio, and you'll see this start to decline. So that's another reason to know these terms, CD8 and CD4, because hopefully you won't run into too many people with AIDS anymore. But if you do, then you'll understand the data that they're collecting from these people. So as you can imagine, if you get rid of your helper T cells, what you're going to end up doing is you're going to end up de-amplifying your innate. You won't be able to defend yourself very well with your innate response. You're not going to be able to do this at all, and you're going to get a decrease in the CD8 ability to activate CD8 cells, and you're going to suppress or decrease in the natural killer cell. So all aspects of your immune system are going to be muted. They're not going to end because, of course, there's lots of redundancy in the immune system. But that's why you see symptomatically in AIDS patients, you see them getting diseases that a healthy person or a person with a healthy immune system would be able to easily prevent from becoming symptomatic. So things like pneumocystic pneumonia that kills or had killed many AIDS patients, is something you see, an organism you often see in the lungs of healthy individuals, but it doesn't cause pneumonia in them because their immune system's strong enough to prevent that from happening. So let me see. We only have a couple minutes left, but could you also talk about the Treg more? Although I've covered it in part.
Yes. So T regulatory cells dampen the immune system, and they can do this either by direct contact or by releasing inhibitory cytokines. So they prevent autoimmune reactions because they suppress self-reactive lymphocytes in the outside of the lymphoid organs. On the other hand, memory CD4 T cells are descendants of activated B cells, which are generated during initial immune responses. They can exist in the body for years, and this basically enables the body to respond faster and more efficiently to later encounters with the same antigen.
Okay, great. Perfect. Okay, so thank you guys. I think the next group, did I even assign the B cells? Is there a group out there still that didn't get to speak? Do you have a spokesperson? Maybe we got through everything I wanted then. All right, so when we come back,